A very good morning, friends. Today we will discuss about an important topic in biochemistry, that is gluconeogenesis. We will discuss this topic under the heading of definition, site for gluconeogenesis, important points regarding gluconeogenesis, then 4 different substrates which are used for the gluconeogenesis, ATP utilized and regulation. So, what is the meaning of gluconeogenesis? By definition, it is a formation of glucose from non-carbohydrates precursors. It is a formation of glucose from non-carbohydrate precursors. These non-carbohydrate precursors, it includes lactate, glycerol, glucogenic amino acids and propionyl CoA. Coming to the important aspects of gluconeogenesis, it is not a complete reversal of glycolysis. The 3 irreversible steps in the glycolysis, they are reversed by the 4 important enzymes. Then 6 ATPs are utilized per molecule of glucose synthesized. Then it is a source of glucose after the depletion of glycogen stores. That is, it is a source of glucose in starvation. After depletion of glycogen stores, which occurs near about 16 to 18 hours in post-absorptive state. Glucose is an important source of energy for the brain and matured RBC. So this pathway is important. Coming to the 4 important substrates for gluconeogenesis, or they are also known as non-carbohydrate precursors. There is a question in university exam. Describe the formation of glucose from the propionyl CoA or lactate or glycerol, or describe the synthesis of glucose from a glucogenic amino acids. So these are the 4 substrates for gluconeogenesis, that is lactate, glycerol, propionyl CoA, coenzyme A, and glucogenic amino acids. Remember, only leucine is exclusively ketogenic amino acid. Then remaining all are glucogenic amino acids. So these are the 4 important substrates from which the glucose is synthesized, that is known as gluconeogenesis. Site. This pathway occurs in the cytosol as well as mitochondria, and important tissue is the liver, and to some extent it is in the renal cortex. So these are the important points regarding gluconeogenesis. Coming to the steps in the gluconeogenesis, the rest of the steps are similar as that of the glycolysis, but these 3 steps are reversed by the 4 important enzymes. Coming to the common pathway, that is the synthesis of glucose from the pyruvate. Then we will discuss the synthesis of glucose from lactate, glycerol, propionyl CoA and glucogenic amino acids. These three steps, they are reversed by the four important enzymes. This step, that is conversion of phosphoenol pyruvate to the pyruvate, it is a irreversible step in the glycolysis. It is reversed with the help of two important enzymes. In the first, this pyruvate is converted into oxaloacetate, and this oxaloacetate is converted into phosphoenol pyruvate. So this irreversible step in the glycolysis, this irreversible step in glycolysis, it is reversed by the two important enzymes. The first one is the pyruvate carboxylase. This is a carboxylation reaction, and remember, the ABC are required for carboxylation reaction. The first thing is the ATP, then biotin and CO2. These three things are required for the carboxylation reaction. By this carboxylation reaction, this pyruvate is converted into oxaloacetate. Then this oxaloacetate is converted into malate. Malate transported out of the mitochondria, and malate then again converted into oxaloacetate, and then oxaloacetate gives phosphoenol pyruvate with the help of enzyme phosphoenol pyruvate carboxykinase. This is important enzyme. This is also an important enzyme. So this phosphoenol pyruvate carboxykinase, there is removal of CO2, and this reaction requires GTP. So this irreversible step, that is conversion of phosphoenol pyruvate to pyruvate, it is reversed with the help of two important enzymes, that is pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Now this phosphoenol pyruvate will undergo the reversal of glycolysis up to this step, up to the synthesis of fructose 1,6-bisphosphate. Then from this fructose 1,6-bisphosphate, there is formation of fructose 6-phosphate with the help of enzyme fructose 1,6-bisphosphatase. It is isomerized to glucose 6-phosphate, and this glucose 6-phosphate is converted to the glucose molecule with the help of glucose 6-phosphatase. There is MCQ on this part. This enzyme is absent in the muscle, and it is also absent in the adipose tissue. It is only present in liver and kidney. That's why gluconeogenesis, or there is liberation of free glucose in the liver and kidney, or gluconeogenesis takes place only in liver and kidney. This enzyme, that is glucose 6-phosphatase, it is absent in the muscle and also in adipose tissue. So these three steps in the glycolysis, three irreversible steps in the glycolysis, they are reversed by these four important enzymes, that is pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. So this is the common pathway for synthesis of glucose from non-carbohydrate precursors. Now coming to the synthesis of glucose from the lactate. This lactate is converted into the pyruvate with the help of reversible reaction. The enzyme required for this step is the lactate dehydrogenase. In this step, this NAD is converted into NADH, and this pyruvate then converted into oxaloacetate. Oxaloacetate is converted into phosphoenol pyruvate, undergoes the reversal of glycolysis. This fructose 1,6-bisphosphate is converted into fructose 6-phosphate, glucose 6-phosphate, and then to the glucose. So this is how lactate is converted into glucose. This is a LAQ in university exam. Describe the synthesis of glucose from the lactate. Now coming to the next, that is synthesis of glucose from the propionyl coenzyme A, that is the odd chain fatty acid. From this propionyl, even chain fatty acid does not give glucose, but odd chain fatty acid, they are responsible for synthesis of glucose during the period of starvation. The propionyl coenzyme A it is converted into methyl malonyl coenzyme A, and this methyl malonyl coenzyme A, it is converted into succinyl coenzyme A, and this succinyl coenzyme A is a intermediate in the TCA cycle. It is converted into the oxaloacetate, then oxaloacetate is converted into phosphoenol pyruvate, and phosphoenol pyruvate undergoes the reversal of glycolysis and gives the glucose molecule. These two steps are very important. First, this is propionyl CoA to methyl malonyl CoA with the help of propionyl CoA carboxylase, that is propionyl coenzyme A carboxylase. This reaction is important as it is a carboxylation reaction. It requires ATP, biotin, and CO2. A, B, and C and carboxylase is required for the carboxylation reaction. Now this methyl malonyl CoA, it is with the help of methyl malonyl CoA mutase. This is again an important reaction and it is B12 dependent. So these two reactions are important. Then the succinyl coenzyme A undergoes TCA cycle, gives oxaloacetate converted into phosphoenol pyruvate. This phosphoenol pyruvate is converted into fructose 1,6-bisphosphate, which is converted into fructose 6-phosphate, isomerized to glucose 6-phosphate, and again converted into glucose molecule. So this is a synthesis of glucose from propionyl coenzyme A. Now coming to the synthesis of glucose from the glycerol. This glycerol, it is converted into glycerol 3-phosphate, and this glycerol 3-phosphate is converted into dihydroxyacetone phosphate. Then it is converted into fructose 1,6-bisphosphate, fructose 6-phosphate, glucose 6-phosphate, then glucose molecule. So glycerol kinase, with the help of glycerol kinase, it is converted into glycerol 3-phosphate, and with the help of glycerol 3-phosphate dehydrogenase, it is converted into molecule of dihydroxyacetone phosphate, which isomerizes to the glyceraldehyde 3-phosphate and gives the fructose 1,6-bisphosphate and ultimately glucose molecule. So this is the synthesis of glucose from the glycerol molecule. Now coming to the synthesis of glucose from the glucogenic amino acid. The important amino acid for the synthesis of the glucose molecule is the alanine. Alanine is the most common amino acid which undergoes the gluconeogenesis. Now the fourth substrate, that is glucogenic amino acids. For UG students, remember the all amino acid except leucine gives the glucose. For the PG student, remember the amino acids which gives the intermediates of TCA cycle. For example, pyruvate, it is given by Pyruvate, it is given by mainly alanine, then serine, glycine and cysteine. Then succinyl coenzyme A, it is given by valine, methionine, isoleucine and threonine. Then alpha ketoglutarate, it is given by the glutamate, glutamine, proline, histidine, then fumarate, it is given by tyrosine phenylalanine, and oxaloacetate it is given by the aspartate and asparagine. So these are the different amino acids which gives the intermediate of TCA cycle, that is alpha ketoglutarate, succinyl coenzyme A, fumarate, oxaloacetate, and it also gives the pyruvate. The most important amino acid, glucogenic amino acid, is alanine, which is used for the gluconeogenesis. This is again an important long answer question. Describe the synthesis of glucose from non-carbohydrate precursor, that is glucogenic amino acid. So there are 4 important LAQs on this topic, that is synthesis of glucose from lactate, propionate or propionyl coenzyme A or short chain fatty acid. Third one is the glycerol, and fourth one is the glucogenic amino acid. Now coming to the regulation of gluconeogenesis, the main key enzymes for the regulation of gluconeogenesis, these are the 4. These 4 enzymes are main, that is pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase and glucose 6-phosphate. These are the key enzymes in the gluconeogenesis. These enzymes are regulated by 3 mechanisms. Regulation. First regulation is the induction and repression. This pyruvate carboxylase, it is repressed by the insulin. So insulin inhibits this pyruvate carboxylase. Insulin favors the glycolysis but inhibits the gluconeogenesis, while glucagon and glucocorticoid, they induces these gluconeogenic enzymes. Second is by the covalent modification of enzyme, that is by the phosphorylation. This by covalent modification, with increasing the cyclic AMP, this cyclic AMP increases or activates the cyclic AMP dependent protein kinases. They phosphorylate these key enzymes in the gluconeogenesis and activates these gluconeogenic enzymes. So this is the second mechanism of regulation. And third, it is allosteric regulation. The 2 important, that is acetyl coenzyme A, it is a allosteric activator of this gluconeogenic enzyme, or it is a allosteric activator of pyruvate carboxylase. And second, fructose 2,6-bisphosphate, it is a allosteric inhibitor of pyruvate carboxylase. In this way, gluconeogenesis is regulated. 6 ATPs are utilized per molecule of glucose synthesized in the gluconeogenesis. So this is all about the gluconeogenesis. Hope you enjoy the video of gluconeogenesis. There are 4 important LAQs related to this topic. Also, there are number of MCQs from this gluconeogenesis topic. It is the synthesis of glucose from non-carbohydrate precursor and important in the starvation process.